name is Dominic, and this is Science with Dominic. In this video, we'll be discussing DNA replication. DNA is the ultimate director of cells as it codes for our traits. When cell division occurs, the daughter cell that is created needs DNA, which is when DNA replication comes into play. There are a few key enzymes that play significant roles in DNA replication. These include helicase, DNA polymerase, primase, and ligase. First of all, helicase unwinds the two strands of DNA, unzipping the two strands of DNA by breaking the hydrogen bonds that hold the DNA strands together. DNA polymerase is the builder of DNA replication. It replicates DNA molecules to help them build a new strand of DNA. Primase is used to help DNA polymerase by guiding it to where it should start working through something called a primer. Primase is the enzyme that creates the primer. This is why primase is known as the initializer. Finally, ligase helps glue fragments of DNA in order to form new strands of DNA during replication. When it comes to DNA replication, it's important to first understand the structure of DNA. DNA is made up of two strands of nucleotides. The nucleotides of each strand form hydrogen bonds with each other. These nucleotides have nitrogen bases that include adenine, thymine, cytosine, and guanine. When they bond, adenine goes with thymine and cytosine goes with guanine. Another important detail is that these strands don't travel in the same direction as they are anti-parallel. When speaking of DNA direction of movement, we say 5' to 3' or 3' to 5'. What you need to know for DNA replication is that the leading strand of DNA moves in the 3' to 5' direction. The lagging strand moves from the 5' to 3' direction. Now let's move on to the actual step-by-step process of DNA replication. DNA replication begins at the origin. At the origin, helicase begins to unwind the two DNA strands. In order to keep these strands separated, single-stranded binding proteins bind to the DNA strands and keep them separated. Primase then makes a primer on both strands, which allows for DNA polymerase to begin building a new strand in the 5' to 3' direction. DNA polymerase adds new bases to the 3' strand as it moves in the 3' to 5' direction. As DNA unwinds, because DNA polymerase can only build new DNA in the 5' to 3' direction, the lagging strand lags behind. Because of this, the new DNA replicated in the 3' to 5' direction is fragmented into fragments called Okazaki fragments. Because of this, ligase is needed to seal these fragments together. At the end of replicating, there are two identical double helix molecules. The two strands each contain one original strand and one new strand. Well, that was a quick review of DNA replication. If you enjoyed the video, make sure to like, comment, and subscribe. I'll be making more AP Biology review videos soon.